My name is Steve Scher, and I'm wearing the hat of the director of the McLaughlin Center today, so I'm not allowed to actually voice my opinion in pro or in con of the question of the session, which is PRS useful, or I'm not sure what the question is, but I'll do that. I'll voice my opinion anyways. And my answer is, or my response is, yes, if you do it properly. And I also want to say I just got an email, just literally 5 minutes ago in my inbox, from the CLSA Magnolia new user account. And we've been waiting for this approval for, I'm not sure how long, but I don't think it's a coincidence that just came in today. But we're very, very happy. So you have a deliverable from your workshop, Lisa, that it's sped up the process of getting approvals. Okay, anyways. So the McLaughlin Center is the largest endowment at the University of Toronto, and I've been the director for about 10 years now. And we support research and education in what we call genomic medicine. And we started to see a real shift in the depth of genetic studies over the last, I would say, 7 years or so, moving right from the amount of data going in, even from clinical case reports across population studies, including genome-wide data sets, common risk variants, you'll hear a lot about in the rest of the session, rare genetic variants, and the impact to scan entire genomes is moving into the clinic. And here at Sick Kids, the impact on rare diseases, of course, is probably most prominent because 70% or so of the kids who come into the hospital here actually have a genetic disorder of some sort or another. So anyways, about 5 years ago, we started to think about where we could have a big impact with McLaughlin Center funding. And we wanted to start to invest in gathering whole genome sequence or genotyping data sets. And then it became very apparent that we needed to have the expertise to analyze this complex data in the most robust way. So we developed a concept, I think it was now 3 years ago, of having an RFA around the concept of polygenic risk scores, and an application came together from this group of individuals. And I won't go through all of them here, you can look at their pictures and names, but Lisa and Andrew will be represented in the session today. And the idea of this group was to develop a critical mass of methodologists motivated by PRS-relevant data and implementation challenges. And the investment really built on the long-standing CIHR stage training program here at the University of Toronto, which many of you are involved in. And the idea here was to give the McLaughlin PRS team funding to address these 5 aims. And I'm just going to read through these so you know the type of research that they're performing here. And if you have any specific questions, you can contact Lisa or Andrew or Leigh or other people that you know in the audience who were on the last slide. And the first was to generalize the polygenic risk score analyses. So to generate one general statistical approach that can be used to include different types of data and that is transferable across populations and contexts. These technologies are very, very early in their development, and they're really being based on, I think, some rudimentary models, and the idea is to make this much more integrative across the board. Secondly, to develop better models based on the new diverse data sets, including omics, clinical, and environmental data. Environmental data we heard from Jeff earlier this morning. Third, investigate PRS in different contexts, relates to population characteristics, ethnicity, population age, we heard from Parminder, and other things. Fourth, to crack the black box, develop a unified platform designated to address the demands from a broad range of PRS users with varying levels of analytical background. I think for many of us in the audience that are trained in human genetics, in particular, and also in developmental biology, where we're starting to use these approaches, we really have to understand what's going on within that black box. And to help do that, the fifth point was to develop a core facility component that would allow to advise on proper study designs, analyses, and implementation, and even into publishing the data. So I underline this because I think this is an important deliverable from the grant, and Lisa and Andrew have developed, and Franz and Leigh and others have developed a group link to the TCAG Statistical Genetics Group here to help provide core service. So if you're interested, you can talk to them. The specific topics that will be tackled are develop unified mixed effect regression models, study the X chromosome, mitochondrial DNA, DNA methylation, time-varying internal and external exposures, improve instrumental variable estimates, precision for causal inference. PRS is a background risk for whole genome sequence studies where rare variants are ascertained and studied. And I think, again, from my perspective, most importantly, as we really do now have the ability to move into whole genome sequence era where you get essentially all of the variants, the combined rare genetic variants and the common genetic variants, is to develop combined common and rare variant modeling. And I think that will be a real strength of the Toronto community because we have access to this data and the ability to actually translate it. Okay, so this is the crew you're going to be dealing with for the next 2 hours. Andrew Patterson, who's here, will be the moderator. And we're very, very fortunate to have, really I would say the 3 leaders in the province of Ontario to join us this afternoon in human genetics and polygenic risk applications. And these include Stephen Neyrod from Women's College at the University of Toronto, Ruth McPherson from the University of Ottawa, and Rob Hagley from Robarts Western University. I'm not going to introduce them because they're going to tell you a little bit about their science and how they're using this data. They'll introduce themselves. And the format of the debate is Dr. McPherson will come up and give us a short introduction of what polygenic scores are and how you use them. And then each of the 3 invited speakers will give a 10-minute presentation on their work. Ruth will go first. She'll stay up here. And then the participants will field questions using the system we've been using this morning, and that will be moderated by Andrew Patterson. There will then be a question period where I guess the most informative questions, and Andrew will probably have his own, will be asked to the participants, and then there'll be, I think, probably a half an hour or so discussion on these questions. Okay? So the debate format, again, is to use the same Slido approach. I think it seems to be working quite well. I suggested to Lisa there's probably some ascertainment bias coming here. But if you have a burning question, you can put up your hand too and participate that way. Okay, anything else? So we'll call the participants up. And Ruth, you're going to go first, or do you want to have the people come and sit at the front table? Later, okay. So Ruth McPherson from the University of Ottawa is going to give us an overview and then tell us about her research.
Great.
Thank you.
Thank you, Steve. I'm at the University of Ottawa Heart Institute. I'm a clinician and scientist, and over the course of the last decade and a bit, my major focus has been on the genetics of what is really a complex disease, coronary artery disease. And we have carried out studies in collaboration with many, many groups around the world, and in terms of anything you do in complex genetics, you need big numbers. So although we started with small numbers and identified the 9p21 locus, everything that's followed has only come about with the help of many other individuals. So most human diseases, I mean the ones we think about, are directly affected really by 4 things. Age, almost everything, the older you get, the more likely you are to develop coronary disease, to develop various types of cancer, to develop Alzheimer's disease, etc. So age, well, we can't really do anything about it, although there are ways that you can age better than others. The second important thing is genetic risk, and so we're born with genetic risk factors from day one, and of course they interact with everything else. So particularly important here is the environment, and we're now kind of thinking of things like pollution, lifestyle, very important, diet, exercise, all of these, not smoking, all of these things that are important. So we can't really single out one thing as causing a disease, but certainly it is a combination of many factors. Okay, so as many of you in the audience know, most human diseases are under the influence of multiple genetic variants, and these are called SNPs, or single nucleotide polymorphisms. And that's the most common type of genetic variation that exists, and each of us carries millions of these, some of which may have an effect, some may not. So when we think about polygenic disease, I'm talking about coronary heart disease, Steve will talk about cancer, and Rob will talk about various metabolic diseases. You know, we're talking about genetic variants that are pretty common. So allele frequency, 1 to 5% for the most part, although there are some less common variants that can have a very important effect. The ApoE4 isoform, for example, for Alzheimer's disease, the BRCA1 and 2 mutations for breast cancer. And then we have rare variants that can also be important, I suppose, in terms of a common disease like coronary artery disease, familial hypercholesterolemia due to a mutation in the LDL receptor or two other important genes can markedly increase the risk of disease. So our approach has, for the most part, been using genome-wide association studies and then imputing from large databases so we can end up with about 15 million good quality SNPs. More and more we expect in the future to go towards more whole genome sequencing. So as Steve said, being able to take into the count in risk scores not only the common variants, and there are many, but also the less common variants that have a high effect size. So GWAS studies are really quite simple. We have these chips, and they used to cost us $450. They now cost us much less than $100. But these have about 1 million SNPs that tag most of the common variation across the genome. Then by imputation from databases such as 1,000 Genomes and now data we have from QK Biobank, you can get 10 or actually about 15 million SNPs that are really very good quality. So how do we calculate a polygenic risk score? We basically have many SNPs. So the ones that are really highly significant, and genome-wide significance means 10 to the minus 8. For example, in coronary artery disease, we have over 100. And these perhaps have the most important effect, and you can basically tally the number of risk SNPs, and you adjust for the effect size of each. So there are certain variants like 9p21 that increase relative risk by about 25%, but for the most part, these increase relative risk by somewhere between 1 and perhaps 8%. But now we are moving towards a more comprehensive approach. So that allows us to use genome-wide SNPs, and we're not going to a particular p-value cutoff, but really can calculate what Inui is called a meta-genomic risk score comprising almost 2 million SNPs, and that gives even more information. So basically, for everybody in the audience, for whatever the disease you're interested in, and the point to make is that with these genetic data, you can create a polygenic risk score for any common disease as long as the GWAS data is out there for that particular disease and for your ethnicity. And of course, one caveat is that most of the data are from Europeans. There are emerging data sets from South Asians and East Asians. So if you are in the bottom 1% of a percentile for risk, then that's great. If you're in the highest, then of course your risk is absolutely greatest, and people in the middle are somewhere there. So why might this be important? I think there's really two important things. So for many diseases, traditional risk factors are important, but they're moderately predictive of individual risk. So if genetic information can improve risk assessment, first of all, for what I do, preventing cardiovascular disease, we have all sorts of good therapies that reduce risk, starting with statins, ACE inhibitors, and now some of these newer agents. But also for things like cancer, when to screen. For example, when do you start mammography in a patient who is really at much higher risk than someone else based on genetic risk? So I think those are the two things moving forward that may indeed be helpful. So moving to what I work on, that's coronary artery disease. It has an estimated heritability of about 50%. So your risk for heart disease, about 50% is due to lifestyle, so obviously diet, smoking, all of those important things, and the other half is due to genetic factors. What we've learned very, very early on was that there's about a twofold increase in risk if a parent or a sibling develops early heart disease. So that was just sort of a clue that indeed genetics are important. What is important, and these are some of the studies we've done at the Heart Institute over the course of the last many years, a CT angiogram is a really neat way to image the heart. So this was a study where individuals came in and CT angiogram was booked. They actually weren't diabetic and they weren't on any cholesterol medication. So what you can do when you see a patient is you can estimate the 10-year risk based on search engines that are out there, for example, a Framingham risk factor score. So this shows the increased risk over 10 years, so people really high risk, a 30% risk of having a cardiac event. And here we have the total plaque score, so the number of epicardial arteries affected and the amount of plaque in each. So we do find a relationship, and that's great. I mean, but R is 0.5, so it's not really too impressive. But what's really important here is that we do these sorts of imaging studies. We find individuals who seem to have very few risk factors, but a heck of a lot of plaque, a heck of a lot of coronary atherosclerosis. And then you have these other folks who are 2-pack-a-day cigarette smokers. Everything is bad and their arteries are whistle clean. So it tells us that we have to go beyond conventional risk factors. And it would be nice if we could identify these individuals at an early age and start preventive treatment. So over the last decade, with many others, we have now identified 160 loci GWAS significant, but 304 with at least an FDR less than 5%. These cover the whole genome, so you're not supposed to read any of that. And importantly, they relate to a number of pathways related to atherosclerosis, which is the underlying cause of heart disease and stroke. Obviously, lipid risk factors like LDL, lipoprotein A, triglyceride-rich lipoproteins, but a bunch of other things. The vessel wall emerging is very important. Inflammation, innate immunity, obviously there's a buzz there, vascular tone, thrombosis, and insulin resistance. So this, for what we do in the lab, is giving us some new insights into the pathology of atherosclerosis. But getting back to genetic risk scores, the majority of these SNPs are relatively common, so present in 2 to 49% of the population. Individually, most have a small effect on risk, but cumulatively, they can be important. So getting back to this, we create a polygenic risk score, and the early studies, not so long ago, this is from Amit Kira and Sid Kathiresan at Harvard, they looked in 3 prospective studies. This is where you follow individuals prospectively over 15, 20 years. And basically, they created a polygenic risk score for each. And so what you see here is that when corrected for known risk factors, individuals in the lowest risk here in blue versus those in the highest risk, there's at least a 2-fold increase in relative risk. So that is clearly important. And you could say lots of things increase risk by 2-fold, but 2-fold is really quite a lot. So more recently, other groups using basically the whole genome. So this is from Thiерson. Here, 6.6 million SNPs. Then you can create a more robust genetic risk score. I won't say it's a hugely more robust, but it is. And then when you look here, what you see is individuals, obviously, as the genetic risk score goes up, risk goes up, and that's sort of an exponential curve. But individuals in the top 1% of a polygenic risk score for coronary artery disease have a 5-fold increase in risk. So that is really enormous. And I think if I was that individual, I'd want to know about it and know about it early. The other point is that there is an interaction between genetic risk and the environment. So this is rather a nice study from George Hindi and colleagues. So these are individuals, this is a polygenic risk score, low, medium, or high. And these are individuals who've never smoked in blue versus those who are currently smoking or had been smokers. So even if you're low risk, if you're a smoker, you're really in trouble, and I think we all know that. If you're in high risk and you've never smoked, you're obviously not like this, but you're much lower risk, and there is always an important fact of smoking, and here this really was a statistical interaction. Getting back to the paper by Kira, so for these prospective studies, all 3 of them, here, once again, you see those in low, intermediate, or high risk right across the board, but they calculated favorable lifestyle. This is based on a whole bunch of different factors that are important in terms of a healthy lifestyle. So once again, those in the high-risk category, if they actually have a pretty healthy lifestyle, then the increased risk is not nearly as dramatic as if they have a lot of genetic risk factors as well as an unhealthy lifestyle. So perhaps we can use these data to motivate individuals to make lifestyle changes earlier. So just to summarize, traditional risk factors for heart disease explain about half a risk. and particularly coronary disease, largely due to multiple common variants, each with a small effect size. Individuals in the top tertile of polygenic risk score, a twofold increase in risk. The top 1%, a fivefold increase in risk. So I think those with high genetic risk are going to benefit most from early intervention, preventive therapy, and perhaps this is going to be, if everything comes to fruition, this is something we'll be able to do in the clinic for relatively low cost. And this is my final slide, just to emphasize how important this is. This is Rory Collins, who's principal investigator and chief executive of the UK Biobank, which is collecting 500,000 individuals who eventually, with the help of Big Pharma, they're actually going to have whole genome data on at least half of these individuals. But even based on the sort of polygenic risk scores that we can calculate now, when he was asked, what is the most exciting thing happening in genomics right now? And he says, the opportunity to implement polygenic risk scores in healthcare systems, something that could have a really big impact on the way we provide public health, potentially cost neutral in that it could enhance the efficiency of screening by targeting those at high risk earlier and those at low risk later, or using preventive treatments more efficiently. Okay, I'm going to stop there.
Thank you, Ruth, for a great introduction. The next speaker, who has quite a steep mountain to climb, is Steve Neyrod. He's going to tell us about polygenic risk in another setting. Just stand in.
Can you go higher? Thank you. Very good introductory lecture. I think makes some very excellent points. I'm going to start slightly off topic. This is a study we've done with my colleagues here, Kelly Metcalf, Vasily Giannakis, at Princess Margaret Hospital, looking at newly diagnosed women with breast cancer. And Vasily took all these patients and calculated their chance of dying of breast cancer within 20 years on the x-axis. And Kelly interviewed them with a text for a questionnaire for anxiety and distress on the y-axis. And you see there's an enormous, this is right after they got the diagnosis. You can see there's an enormous variation in the level of anxiety. And you can see there's no correlation between the level of anxiety and the probability of dying, which ranged from 2% to 60%. Which is sort of irrational in the sense that the risk didn't correlate with the effective response to the risk. Now, perhaps that's because the patient's risk, their perceived risk, wasn't the actual risk. Perhaps because there wasn't proper communication between the doctor and the patient. Or perhaps it's because affect, which is the emotional response to information, is dichotomous and it's not related to a continuous variable. And I think that's often the case, particularly for cancer, where the response is more you have it, you don't have it, you have the risk factor, you don't, and very difficult to translate that into a continuous variable. So what determines the behavior? The behavior to take a prevention, the behavior to go for screening, the behavior to have surgery. Is it the actual risk, the perceived risk that the woman has, or is it the affect generated by the knowledge that she has the risk? So when we talk about, we got a good introduction to SNPs and personalized risk scores. I'd sort of look at the following caveats that for these to be useful, the following things should be shown: that there should be a genetic assessment based on personalized risk scores, SNPs, which can divide the population meaningfully into those at low risk and high risk, and that the cutoff is meaningful. The test should be widely available and acceptable to the population. The information regarding the test, the meaning of the test, the interpretation, and the possible interventions that could proceed should be communicated clearly and meaningfully in a way that the patient understands. We need to have an effective intervention for those who test positive. The intervention should be acceptable to the patient, and the uptake rate of the intervention should be higher among those who test positive than those who test negative. These are sort of a population perspective on how to introduce a test into a population and what to expect from it. So in the first one, let's look at the SNP has to generate two groups. So Vasily and I took every woman in Ontario, 6,301,000 of them, and generated, using personalized risk scores that were published in JNCI, and using a distribution of risk factors and age, generated their lifetime risks from today until age 80 for them to die of ovarian cancer, and we plotted that. And what we can see is nobody was high enough risk for an intervention. Except for the BRCA1 and 2 carriers, there's no person that was 5% risk. And 5% risk is what we consider to be high enough to validate a preventive surgery. Preventive surgery is a way to prevent ovarian cancer. The screening doesn't work and there's no chemo prevention. So the fact that even though you had 6 million women getting SNPs, nobody got preventive surgery except those that were scared of it, the SNPs didn't contribute. And that's generally because their disease is rare. If the disease is only 1.4% in the population, I can multiply that by 3 and I only get 4.2%. So the disease is too rare. No possibility of SNPs currently to help us determine who should get a preventive surgery. A second thing, the information of the test should be easily communicated and understood. Now, this is a test study me and Kelly did a few years ago. 2,000 women got a genetic test for BRCA1 mutations, Jewish women, Women's College Hospital. The patient thought their risk was 36%. These are 2,000 women on average. The computer said it was 11%. So perceived risk and actual risk are completely different. And when we actually did the BRCA1 test on them and tested them a year later, what we found is those women, we told them their risks and we sent them a letter, and those risks were 11%. A year later, they reported back their risk was 33%. It didn't go down. How did we communicate it? By genetic counselor and by letter. And this is one's really interesting. We did the same test. We gave them a risk of ovarian cancer, and we told them you're positive or negative. The women that were told you had a mutation were distressed. They gave up with a high risk. They said their chance of having ovarian cancer was 26%. That caused them a lot of distress, and 88% got an oophorectomy within a year. The women who had a negative test were not distressed. They said, my risk is 26%. And they were not distressed, and none of them had an oophorectomy. But they both said it was 26%. So what options do we have? So in breast cancer, we have preventive surgery, we have chemo prevention, and we have screening. These are sort of the tools of the trade that we can apply to the population using personalized risk scores. Next, the uptake rate of the intervention should be higher in people who test positive than people who test negative. This is a study done by Alison Curran recently, where she tested, I don't know how many hundreds of women for common genes, BRCA1 and 2, rare genes, and came up with the following results. And so she tested all the women for BRCA1 and 2, found 64 mutation carriers. And then these are people with breast cancer. Of those that had a mutation, 79% appropriately had a bilateral mastectomy. But of those who had a mutation in one of those other genes listed on the bottom, 38% had a bilateral mastectomy. Of those who had a variant of insignificance, 30% had a bilateral mastectomy, and those who had a negative test, 35% had a bilateral mastectomy. Therefore, the uptake was not more common in the individual who tested positive than who tested negative, with the exception of the BRCA1 and BRCA2 carriers, who I've been recommending testing those women for 20 years. The question is, by doing a personalized risk score, where's that going to fit? Well, I'll tell you, 35% of them will have a bilateral mastectomy regardless of the personalized risk score, and that's never going to change. Next, the intervention should be acceptable to a high proportion of clientele who test positive. So we asked my patients with a BRCA1 mutation, high risk, 4,000 women with a BRCA1 mutation, to how many took tamoxifen, the only chemo prevention approved for BRCA1 and 2? 7%. Why didn't they? 7%. They took the only thing that's out there, and they didn't take it because they think they say it's due to side effects. In reality, it doesn't relieve distress. The affect of a woman, the sensation, the distress and the anxiety of a woman is not diminished by tamoxifen. It is diminished by surgery. The day the woman has her surgery, the next morning she gets up and feels an enormous sense of relief. chemoprevention offers no sense of relief, and they won't take tamoxifen. The intervention should be effective for those who test positive. So let's go back to mammography. So we offer mammography to women. Okay, so this is basically the evidence. At Women's College Hospital, I've been there 25 years, and when I got there, we collected information on everybody with breast cancer, and we followed them for 20 years. And when I compare the women who had their breast cancer diagnosed by mammography, non-palpable mammogram detected on the top, and palpable, they could feel the cancer on the bottom, we see this enormous survival benefit. One had a 83% 20-year survival, one had a 60% survival, which of course shows a tremendous benefit for mammography. If we stop there, but then Tony Miller and I ran a randomized trial of 90,000 women in Canada, the National Breast Cancer Screening Study, and we randomized 90,000 women to mammography, half of them to mammography, 5 of them, and half of them to usual care, and counted the deaths over 25 years, and there was no difference. That's a randomized trial. So on the left is the observational trial, which shows this enormous benefit of mammography. On the right is a randomized trial, which shows no benefit. Well, which one do you accept? Well, the one on the left, of course, the observational study, because that's the one that you have to accept that if you're going to believe personalized medicine. Anybody who in this audience who's going to accept the idea that you can reduce death from screening mammography has to accept data that's not from a randomized trial, because that's the only one that fits the paradigm. So let's assume that we can prevent 40% of the deaths. Let's take 10,000 women who have, and give them a personalized risk score. Assume 10% of them go for the test. That's 10,000 women. Assume 10% of them are in the top decile, and the risk is tripled. So those women have a 3% risk of breast cancer between the age of 40 and 50. So they go for screening at age 40 instead of age 50. We would then expect to find 24 cancers in that group between age 40 and 50. Based on that 3% of 800, we would expect 20% to die, or 4.8 women to die. I can reduce the deaths by 40%, which is extraordinary. So I've prevented 2 deaths. Unfortunately, 1,998 of them died anyways. So introducing a personalized risk score for genetic testing in the province of Ontario is great. It'll prevent 2 out of 2,000 deaths. But that's the best we can do. Anyway, so I leave it to you. These are just observations from my own research over the last 25 years, which led me to think that there's more to it than just the personalized risk score, that there's all the issues of how do patients react, what are patients looking for, affect, et cetera, which we have to consider before we can expect to see any impact at a population level. Thank you.
And our last speaker is Rob Hagley.
Thanks, Andrew, and thanks for the invitation to speak to you. So, I take care of patients. I also do research. My patients I take care of are also my research subjects, and so there's the interface of reporting research results back to them, and then some insights from seeing that. So this is a patient, been my patient for a while. He's got a high LDL cholesterol. Both his parents have had dyslipidemia. Maternal uncle had bypass grafting at 41. He was intolerant to a statin. He himself developed angina and needed bypass surgery at the age of 45. He then was started on one of these injectable drugs for cholesterol, evolocumab, a PCSK9 inhibitor. So, LDL came down to 1.5, so he had a great response. He had clinically probably, it looked like it was a monogenic form, and with that family history, it looked like it was monogenic, but when we then ran our targeted panel on him, which looks both for monogenic, and we also designed the panel to microsequence the SNPs that we use to build the polygenic score for cholesterol, it turns out he had a high so this was actually polygenic high LDL that appeared at the clinical level like a monogenic condition, and actually equally as severe. So in terms of at the the clinical decision making, it was essentially like monogenic familial hypercholesterolemia. So you're a nice group. I will skip over that. The reason that we know about how lipids are metabolized is largely through human genetics, largely through monogenic conditions. And so we have LDL particles. I don't know if this projects. Oh yeah, you can see it there. Oh, that didn't sound very good. Oh. LDL, which is this, that's the bad cholesterol, that's the particle here. And then also I'm going to be talking a little bit about triglycerides. So I've been, so even going back to like 25 years ago, we know that the lipids out in the general population are a polygenic trait, the archetypal quantitative polygenic trait. So this is then when we were using these very primordial methods, TACMAN genotyping, looking at one gene at a time. Of course, Ruth showed that we can now look at 6 million SNPs on a microarray, but here this was like a chemical thing that was like really painful to do for each one. And then so this is actually multivariate regression analysis, partial R squares that are then breaking down the proportion of variation of the specific lipid trait that was due to a particular SNP or single nucleotide polymorphism. And so fortunately in those days, we didn't have many to look at. We maybe had like 20 to look at. So then, but this at least established the point that these traits, for example, LDL cholesterol, that the first gentleman that I showed you, it's largely polygenic. We could even in those days with a sample of 1,000 people show the contribution, and these are small effects. So these are common variants, small effects, but cumulatively, when you add them up, the attributable variation actually ends up being quite big compared to other things. And then, same thing, now, kind of 13 years later, hypertriglyceridemia. So I've been like, focused on polygenic, but then, in a different era of technology. And so then when SNPs came around, and SNP microarrays, then this actually fit the paradigm, but then this allowed us to then look at it on a very broad scale. And so this is then for, say, triglycerides, that other continuous variable. And this would be then the distribution of weighted risk scores. So here we were just using small risk scores. Ruth showed that now the sort of de rigueur is to do like 1 million SNPs or 6 million SNPs at a time. So back in the day, we would just choose the top 10 or top 15 by effect size, by beta, and then include those in our risk score, build our panel to include those. And this is really then what you see. This is then the distribution of of risk in red, the distribution of scores in people with normal triglyceride levels, and then in people with like really high triglycerides. So that it was to the extent that the blood looks like milk. It doesn't even look like blood anymore. It's like creamy blood. This is then the distribution of those scores. And when you do the stats on that, it's like 10 to the minus 30 for the difference in the distribution. So it's really clear that biologically these scores are telling you something, and then we put them in bins. You can see on the right-hand side in terms of then I guess these were 7 groups, so septiles. But but on an individual level basis, because there is so much overlap of the score, so when you're out at the extreme, an extreme score, if you're out here, that'll tell you, okay, you're more likely to be hypertriglyceridemic, and then down here the same on the normal side. But in the middle, I mean, there is the middle that overlaps, and this is then the sort of the limitation of these scores in the clinic, because we're reporting results back to an individual patient. So at a population level, like it tells you something about the biology. So here's an example. We do in like 2013, we designed this targeted Illumina panel to look at all possible genetic causes that we knew of in that time for, say, high LDL cholesterol, bad cholesterol. So we chose our top 10 SNPs. These came out of GWAS studies. So the GWAS has identified 100 of them. We basically then chose the top 10 based on the beta. And then we designed our sequencing panel to micro-sequence these SNPs. So the sequencing panel then looks both at the coding regions of the gene and allows us to find rare variants, and then at the same time, with the excess capacity on the panel, we're micro-sequencing the regions around these SNPs, and we're able to derive these common scores. So we can both rare variants and common variants simultaneously. And so then this is a report. So comes back and our ethics review panel. So as I mentioned, every patient that I see is also a subject for my research, has been this way for 25 years. So our ethics panel said, well, when you report the results back to the patient, you should let them know about all forms of variation. So patient comes back for their second visit. Oh, you remember that DNA sample that you gave? Oh yeah, well, what did it show? And everybody's interested. Everybody wants to know what the DNA testing showed. Oh yeah, it showed. And then, a lot of the time the high cholesterol is due to a single big gene that totally explains. That's not what you have. You've got different. These are a bunch of little genes that are all kind of acting together, and they're all, but cumulatively, and you've inherited. So normally we would have like 10 out, like an average person would have 10 out of 20, but you've got 18 out of 20, and that puts you in the top 99th percentile for this trait. And it's as if you had like a single gene that was like skewing your cholesterol level. So your cholesterol is genetic, is just a different way of getting the high cholesterol. So, and then we've now run this panel on, 4,000 people almost. So like the whole, this is just the general population of my patients. So the whole, and this is polygenic traits. So unfortunately, there's no dichotomy. I would love to have, affected, unaffected, the first item on Steve's list of what makes, Steve Neyrod's list of what makes a good, clinical, so here there's the middle, it overlaps, but I still think it tells you something. So for example, patients that come in with high cholesterol, so LDL between 5 and 5.9, 42 are polygenic, and then the high, or monogenic, I should say, and then a high polygenic score in about 15%. And then with the very high cholesterol, like the people who have the extreme of the trait, the majority of those are monogenic and very small number are polygenic. So these are the kinds of things that we can do. We can also say, do they respond differently to these drugs? So we've got these new cool drugs, these injectable drugs that you give twice a month instead of taking a statin pill every day. So the patients really actually like these medications. The only problem is that they're very expensive. So if you have insurance, they're great. So for you have monogenic hypercholesterolemia, reduces LDL by 58%. If you have polygenic hypercholesterolemia, so it looks like it's even on a percentage basis a greater reduction, but you wouldn't hold it back. You wouldn't hold back the medication because the monogenic form on a percentage basis responds slightly less compared to the polygenic form. And so then we can, break down all of our lipid traits. So for example, HDL, the good cholesterol. So for example, LDL is like 53% monogenic, 13% polygenic. Triglycerides is the other way around. It's sort of 17% monogenic, about 30% polygenic, and then the rest we don't know. So this is like the subject for future grants. So thank goodness we can't, not everything is explained by genomics because we need to keep writing grants to fill in the rest. Okay, so here's some observations that I've just observed. I'll go very quickly because I know I'm near the end. This could be my summary slide. So in general, patients are very interested in genetic results. Patients seem to refer, this is the other way, you can do a weighted score where you can take the tally of the alleles that are raising the trait. You can either do the raw tally or you can weight it. So patients prefer the tally. We see clinical differences. It's motivating for patients. Family docs then often have questions about polygenic risk. So far, knock on wood, no adverse ethical or clinical outcomes in reporting these polygenic scores to people, no adverse effects on insurability. Sometimes it helps get drug coverage. And we find that the small scores, at least in the clinic, work as well as these, we think, as these million SNP scores. In fact, I think the small scores are easier for a patient to understand than the big score. Thank you for your attention.
Okay, so we've now entered the discussion section of the afternoon. So both from the audience and from Slido, I'm willing to accept questions that the panelists will respond to. I think they have 2 minutes each to respond to the questions. So perhaps I'll start off. I have a question for Steve Neyrod, first of all. I just want to make sure that I understood your presentation. Am I correct in saying that you said that if we provide breast cancer polygenic risk scores to women, 35% of them will have prophylactic mammography, irrespective of the result?
These patients had breast cancer.
Pardon?
They were patients with breast cancer.
But in the general population, what proportion do you think? would
35,
have-
well, I would say, and we've studied it here in Toronto, this was a study from California, about 25% of women now treated for breast cancer at our hospitals across the board, including DCIS, have a bilateral mastectomy as their initial treatment. And that, what we found seems to be irrespective of the stage. Now, we've just written a 10,000 word article on why, but seems to be a lot of interest in reducing fear of contralateral cancer. Sort of iatrogenic, so women, 1/3 of women respond to the fear of contralateral cancer by having a bilateral mastectomy, but that decision is not related to the risk of contralateral breast cancer.
I see. Do the other panelists want to weigh in on this, or? Let's move on. Rob, I was a bit surprised that patients preferred the unweighted score, because intuitively I would expect a weighted score to behave better.
Right.
So perhaps could you provide some insight into kind of what you think's going on there?
I think it's just the explaining it. So I think they understand the concept better because you can explain it to say, okay, there are 10 genes, so 10 loci, and there are 2 alleles at each locus, and you could be really unlucky, and at each locus, at each of the 10, which are totally independent of each other, you can be super unlucky, and always at every locus, both alleles are raising for the trait, so then you would have 20 out of a possible 20. Or alternatively, you could have 0. You could have at every single locus, you could have a variant that's associated with the lower, lower level of the trait. So that's 0 out of 20. And then it would be and then you just simply tally them up. It's like every possibility in between. Once you put in a weighting factor, and you then, so they can understand the tally, because that's just like counting genes or counting loci or something. But if you tell them, okay, well, it's we're going between 0.3 and 1.98 maximum, then they need to understand more. Then they have to understand mathematics. They have to understand that it's like, it's a continuous distribution. So when I say, I agree. I think the weighted score would discriminate better, actually clinically is more useful. But that's not what I'm talking about. I'm talking about translating the meaning of a polygenic score to a member of the general public who, has high school math. Then the tally is an easier concept.
And Ruth, would you like to chime in on this topic in terms of transmitting information from polygenic risk scores to patients, kind of weighted, unweighted, kind of what are the challenges?
Well, I agree that, when we had the 9P21 paper, we had a lot of individuals who participated in that study that wanted to know what their risk was, because that was sort of big press. I think there will be a challenge in educating individuals about what genetic risk means. And, but, particularly when I have a patient in a clinic, this patient who has early coronary disease, like a guy I saw on Friday, his LDL had never been higher than 3, but his father and brother, early bypass surgery, this guy, early bypass surgery, he really wants to know what his risk is. So I think for some individuals, it's going to be very motivating.
Steve, thank you.
I think you guys, it's all wishful thinking. Take the patient, give them the score, tell them everything you need, you want them to know, the risk score, the number of alleles, their particular level, their risk of this, and then call them back a year later. If you do that and show me that they can recollect the original conversation and reproduce the numbers, I will be a convert.
Yeah, we're doing it. We're doing that study. Yeah.
Perhaps for Stephen. So it sounded as though you were doing genetic counseling by letter in one of your studies. Yeah, you said you returned results to women by letter. I think that was the mutation screening in the Jewish population. Is that right?
Okay, I think we only return, if they were negative, we might have given them by letter. If they were positive, it was, this is standard for us now.
I see.
We do it at the Screen Project. Anybody that's local, if they're positive, we call them up, and usually we give the result over the phone, and then we make a personal appointment. We found that if you give the result at the time of the personal appointment, the distress makes it hard to communicate. We call them, and then they come in the next week. If they're negative, we send them a letter, yeah. Seems to be okay.
Questions from the audience. Jen, did you have a question?
Okay, so I just wanted to, so I guess back to the previous point, we are doing the study where we are in Ontario and Quebec calculating women's risk of breast cancer, communicating that risk to them, and then following them up over time to see if their behaviors change, who they tell, how they feel about it, if they get screened, if they don't. So we will sort of get at your question, Stephen, but to your point of, you know, you need to be able to do something, like have an effective way of preventing disease and have women being willing to do it. I think that's a really good point. We did a population level survey of general population unaffected women across Canada and found that women are very willing to accept more screening if their risk is high or higher than average, but very unwilling to accept less screening if their risk is lower than average. And I think that if we really want to see a way where we can truly stratify women at the population level to have targeted screening, that certainly, they only seem willing to go one way rather than really being able to accomplish it across the spectrum of risk. So.
Well, that's a good point, but why do women like to have mammography? You think women like to have mammography because they believe it reduces the risk of dying of breast cancer. That's not the reason. Women like to have mammography because a negative test has a huge impact on their affect. The women's, it's iatrogenic. We invite them every year. 2 weeks before the mammogram, we make them anxious. They go for the mammogram, they're anxious. They get a negative one, and there's a tremendous surge of relief. They value that. They won't tell you that's what they value. They say we do it because of my kids and because they do it of this. That's what I call the hitchhiker's heuristic. But what they become attracted to is the effective sense of relief of being told you don't have cancer. And it's probabilistically, the posterior probability of having breast cancer with a negative mammogram is actually quite predictive. It doesn't mean that the mammogram reduces mortality. It just means you can predict quite well. If I say to you, you have this, you're worried about something, it may be incurable. But if I test you for it and you don't have it, you're really relieved. That's what medicine's all about. It's not really about helping people necessarily. It's about telling them you're okay because they want to hear that. And they'll come every year for that message.
Ruth, do you have any comments on this?
You know, we're talking about apples and oranges because, I mean, cancer is quite different, and what a woman wants to do after she's had cancer in one breast is one thing. You know, I think the utility of polygenic risk scores is really for these common diseases where we do have good preventive therapies. And coronary disease, we definitely do. Even things like Alzheimer's disease, about 40% of risk is due to common genetic variants. And there are emerging data that if you're at high risk for cognitive impairment later in life, doing all the right lifestyle things can be beneficial. So, you know, my disease is different than Steve's disease. And I think there is a real utility for assessing risk.
Yeah,
And-
actually, I
and
wonder
Rob?
if I could also, so just to expand, I totally agree with what Ruth has said, and just so in our area, just an example, so statin treatment. So statins have been generic for like 20 years, and they lower LDL, but still there is this whatever pervasive feeling people don't want to take them, even if they feel that the statin some people feel that the statin poses a greater risk to them than they're living with their cholesterol. Once they have their heart attack and stroke, and if they survive, and if they go through rehab and they recover, then they're more motivated to take their statin. But obviously we're trying to prevent that. And so what I have found, and what we are now trying to test, is that if they then know that their cholesterol is high for a genetic reason, be it monogenic or be it polygenic, my impression is that they're more likely to comply with their statin treatment. So then I'm using it, I use it in in delivery of healthcare in a preventive sense.
I don't disagree with either of you guys. I think what you're doing on a personal level to personal patients that are coming in with disease is admirable, and I would use the genetics in a clinical context. What I'm sort of talking about, when guys are saying the future is rosy, like you're talking about a major shift and a major paradigm shift in healthcare, both for prevention and treatment, and I think that's make-believe. And the reason is, if you look at Ontario, what percentage of adults over the age of 60 have some kind of hypercholesterolemia, hypertriglyceridemia that require intervention? Those medications, as far as I know, work. The statins work, and that's good. So then we have the polygenic risk score. But the trouble with being in the 99 percentile is only 1% are in the 99 percentile. We're stuck with that. If 10% of people were in the 99th percentile, we'd have a great paradigm. Statistics don't work like that.
Ruth.
But a third of the population is in the top tertile, where the increased risk is twofold. And twofold is quite a bit for disease that's very, very common. This isn't an orphan disease.
And the data that I showed was for the 10th. We used our trade-off that I use is the 10th percentile.
Let's go to the audience for questions. Elise?
Thank you. Thank you to the three of you. The first speaker mentioned that, I have two questions. The first one is that a polygenic risk score could be established with about 50 SNPs. And I was surprised at the small number. I was wondering, what size cohort did you need for that?
So this is for coronary disease.
Yes.
So, as time went on, more and more groups around the world, we got 50 SNPs and then more and more. So the early polygenic risk scores were based on basically the low-hanging fruit. So those were the genetic variants that were most significant individually, had the greatest effect size, albeit individually a small effect size. And as more data have become available, and we're talking about basically thousands and thousands of individuals.
40,000.
40,000 in our recent Nature Genetics paper. So that is really a robust data set. But for polygenic risk score and predictive value, it's not a case control study that's really important, although it's of some importance. It's the prospective studies. That's why the studies, the ERIC study, atherosclerosis risk in communities, diet heart study, and the Women's Genomic Health study, those followed individuals for 20 years. And obviously your genes don't change. So the polygenic risk was predictive for lower to the top tertile, a twofold difference in risk, and statistically extraordinarily robust.
So these
Elise, we'll come back to you.
Yeah.
Okay.
Slido, anonymous, says, if this was used more broadly, would you be worried we are basing the scores on the wrong SNPs? GWAS SNPs from smaller studies do not always replicate.
These are huge studies. I mean, really, yeah. So I guess the most recent meta-analysis is over 100,000 controls and approximately 60,000 individuals of premature coronary disease. Huge, huge studies.
Anybody else want to address that?
Yeah, so for lipids, I think the latest one, that there was like a 1 million person study. So these SNPs. So the thing is, the ones that are at the top of the list are always at the top of the list, whether it was 20,000 or now that they're 1 million. And what we're now seeing is that when you go to 1 million people, there's just now the sample size increases, so the threshold, the genome-wide level of significance. So they're just more and more being added in. And then this whole theoretical concept of then using 1 million SNPs to build a score. Because then 1 million SNPs is then, okay, if we could genotype 1 billion people, then the whole concept of what is genome-wide significant is relative, right? If you had just a big enough sample size, so what becomes more important is the directionality, not the p-value. And so then these million SNP scores, they're just adding all the variants that are moving the trait in the same direction, even if it's incremental, and even if the p-value is close to 1. But you add them together, so and then the increment is something. So when you're using like a SNP score of 50, 50 gives you like an odds ratio, say, of 3, and then you go to 100 or say 10,000 SNPs, it gives you an odds ratio of 3.6, and then you go to like 1 million SNPs, and then maybe you're up to an odds ratio of 4.5 or 5. So you are adding, so it's almost mystical. There is something that you take a bunch of things that are individually not significant, and then you add them together, and then you continue to derive information from the aggregate.
Yes, Steve.
It's an interesting question. I mean, I figure if it takes 1 million people with 1 million different variables to show an effect, it's probably not a very big effect.
Individually not, but in aggregate it is, for sure.
Back to Slido. To what extent can polygenic risk scores be modified by the environment / social stress, etc.? Ruth?
Well, I suppose there are some epigenetic effects, but as I showed in a couple of my slides, certainly the impact of a polygenic risk score is in part contingent on your risk factors. So, I mean, this is sort of part of the argument, because if you're at high genetic risk, but your lifestyle is pristine, your absolute risk is much lower. So I think that is really the most important message. And moving forward, can we convince individuals to make lifestyle changes or take a medication based on the information we give them about personal genetic risk?
Rob?
Yeah, so this is the one thing for sure. For polygenic risk, it is much more amenable to environmental influences, which includes medication, and diet and exercise, and so that's the big hope, is that somebody's got a cholesterol of 8, but it's predominantly polygenic. Then at least you then stand a good chance of intervening with your treatment. But even for monogenic hypercholesterolemia, there is evidence that is amenable to environment as well. And so these studies that have been done in Utah and looking over generations for the same mutation, and different ages of onset of disease, different levels of mortality. So there may be other factors, so environment is still important for monogenic. So that's at least in the metabolic field. So for things like diabetes and cholesterol. For cancer, I would be interested in BRCA1, to what extent then can you modulate that environmentally? But certainly in the metabolic field, for monogenic you can, but for polygenic even more so amenable to an environmental modification.
Next Slido question. How well do PRS translate across various ethnicities?
This is a bit of an issue, because to basically develop these polygenic risk scores, we need genome-wide association studies from very, very large groups. At the moment, the majority of data is from European individuals, and that certainly doesn't apply well to the African population, much older population, much smaller haplotypes, etc. There are emerging data on South Asians. There's been a lot of studies in Pakistan, and there are a fair amount of data from East Asians, from Oriental populations. But yes, this is certainly one of the issues at the moment.
Yeah, so I agree. The polygenic scores mainly that are used is just because of the skew or the like the bias towards initial studies done in European populations. So we really can't use those in other. I mean, and I can't use them in the clinic in other ethnic groups. So it's almost like different geographic ancestries. However that is defined through principal components or through whatever way you define a geographic ancestry, but different, that you would need a different score for different ethnicity or geographic ancestry.
Steve, I don't want you to feel left out. Questions from the audience? Yes, second row. Can you press the button? That's good.
Hey, so I'm coming from a psychology and human factors background, looking at kind of communication and information literacy. And you guys, all three of you touched on different topics of, like in genetic counseling, how people interpret that, how to manage those expectations, how to clarify misinformation. And I'm just curious if you have any go-to strategies, recommendations, even partnerships that help manage that when it's the wrong time to convey that, and also sort of adhere to the patient consent kind of model, just how to balance those two tension points of inducing anxiety versus relieving it.
I think it depends on disease. People are not very anxious about coronary heart disease, so it's kind of easy enough. They just want to hear more about it. I think in terms of cancer, everybody's anxious, and you know, I think the emotional side, the reactions are probably more important. But, you know, I don't know, in terms of basically starting with a healthy population, will this information be so scary that they just sort of totally avoid it, or will it be helpful? I don't know. I'm sure that Steve has some very negative reply to that.
No,
You have nothing to say, Steve?
it's too complex.
It is.
Rob?
It's really complex.
Yeah.
Yeah, well, and so I mean, it's a great point. So part of it is actually then having a genetic. So I think having a genetic counselor working together with somebody who then has that specific expertise that then can sit and explain with the patient, rather than, so it shouldn't always be then the primary healthcare provider that should do that. But I can give you other examples of how people, which I'm sure you've seen. So not even talking about genetic risk, but just talking about, you know, Framingham risk. So you know, we would then go through the algorithm, calculate somebody's risk of a heart attack or stroke, and it ends up being 50% in 10 years, which is enormous. That's huge.
Yeah.
But we tell the person, okay, listen, you got a 50% chance, and then it reflects back on the nature of the person. It's like, oh, okay, well, glass half full, 50% I'm going to be okay. So I, you know, don't need to worry about that, right? But then another person, you tell them exactly the same thing. Oh, hey, no need to worry. Your risk is only 1.1% in the next 10 years. And then they can't sleep at night. That's like, they want their risk to be 0. 1% is too high. So a lot of it then, so even like whatever we think we are conveying in terms of like, you know, translating our understanding, that's less than half of the equation. So much of it is how the patient absorbs that.
Very true.
Next question from Slido. Does polygenic risk account for interaction effects between SNPs? Are we missing significant effects if we're not accounting for these interactions? Ruth?
Yeah, so we've done a little bit of interaction analysis and published a couple of positive studies, but you know, statistically that becomes extraordinarily difficult, particularly given the size of the data sets. And we tend more to look within pathways. So one interesting way to look at coronary disease and the genetic information that we have is, even before we had these sort of genome-wide genetic risk scores, you start to see that you have genetic variants kind of weakly associated with a trait, but if they cluster within a pathway, that's really telling you that pathway is important. So that's not exactly gene-gene interaction, but in a sense, it's putting those genes into a pathway. So yeah, SNP-SNP interaction, statistically very, very burdensome, and I think, yes.
So yeah, just to, I agree. So really what we're, like all of these significant associations come out of linear statistics. It's like regression analysis, like, using linear regression. So to the extent that biology follows linear models, but then, so yeah, of course, but then the thing is that we don't, so we barely know what we're looking at. We're just like capturing a little bit of the variation by trying to fit, an exponential square into a round hole, or whatever the expression would be. But yeah, like biology is not linear, but so far, everything has been derived, or 99% of it has been derived with linear statistics, which fails to capture it. So yeah, I agree. If we then start looking at interactions or even higher order, exponential relationships or something that's not linear, we're gonna capture more of the variation.
Steve, do you have thoughts about interactions? No. Pass. Okay. Back to Slido again. In the US, insurance companies use genetic testing to assess insurance premiums. Will Canadians with high genetic risks be at risk for increased premiums?
Yeah, so this was addressed. So in the United States during the Obama era, they had the GINA, the Genetic Information Non-Discrimination Act. So you could argue, how well that works or doesn't work. So in Canada, there was something similar in the first mandate of the Trudeau government about 2 years ago that was put in, that was meant to have the same sort of impact. I think it may be less of an issue for medical coverage in Canada. I think it's like just a totally different ball game, thank goodness, in Canada. For life insurance, that might be different. But so far, I have not seen any. Have you seen any?
No, it's a good question.
Ruth, anything to add?
I think people are protected in a sense, polygenic risk, because it's all of those variants. I think that's less scary than having an insurance company do your ApoE genotype and decide if they're going to cover you for later in life. I mean, it's really important in terms of the high effect size variants.
Ruth, you talked about Framingham risk scores. How do polygenic risk scores stack up against conventional risk scores? Should they be combined? What are your thoughts?
Yeah, so the data that I showed you, for example, the simple thing, highest tertile twofold increase in risk, that was corrected for conventional risk factors. And the whole problem with the Framingham risk factor score, I think I showed you the slide from the CT angiograms. I mean, our correlation was, explain 25% of variation in plaque volume. So Framingham risk factor score is a place to start, but boy, you can't stop there.
Yeah, so just another comment. Again, I always see Ruth was my student many years ago, so I cannot help but
He's much older than I am.
Yeah, one of the best clinical clerks, I think, at Toronto General. So in Framingham, we do have this sort of rule of thumb clinically, that there is, say for example, family history. So a family history of an early heart attack. So that's not officially part of the Framingham score. So Framingham is like, age, sex, blood pressure, cholesterol, diabetes, smoking, and then you put it together and then you can predict, but it's only about whatever, predicts 50% of the possible risk. But family history was never collected, but there's an adjustment that clinicians use. So if dad had a heart attack, say under the age of 55 or 60, you take whatever risk Framingham tells you and you multiply it by 2. That's a clinical rule of thumb. Okay, so and so now what we've been trying to do with all of this, Human Genome Project and next generation sequencing and SNPs and GWAS, is to try to see, because it doesn't cost a penny to ask the family history unless the patient is an orphan or does not know their family history. But so that's a zero cost test. So is any of this any better than just asking the patient? Does it provide an increment over Framingham of just what we're doing as a clinical rule of thumb?
But yes, the studies of the polygenic risk score still significant for CAD when you correct it for family history. Partly because, well, family history is, sort of a coarse measure, and people don't know what their father died of or whatever. They think a valve surgery is the same as a heart attack, et cetera. Sure. But yeah, it's giving more information. But I agree, family history is really important, and I think a lot of primary care physicians don't really ask it often enough or put enough emphasis on it.
And then, of course, family history also includes shared environmental factors as well.
Steve, conventional risk factor models, do you have anything to say about them? I don't want you to feel
Well, there's
left
certainly
out.
a lot. I mean, we do them for breast cancer. They seem to be pretty good. Sort of, the question is currently, and I think Jennifer Brooks is probably working on that with Anna Chiarelli now about introducing SNPs and mammographic density to a model. I used to be quite keen on it, not quite so much anymore. So the Wisdom trial in the United States, there's a big trial run introducing mammographic density, BRCA1 and 2, and SNPs to see if we can improve screening. I think you guys are all have an interesting academic research portfolio. I'm just saying I wouldn't be recommending this, and I wouldn't be making any claims until you could show me that in a trial, a randomized trial, if I took 20,000 consecutive patients and I randomized 10,000 of them to the PRS and the 10,000 to conventional care, it would make a damn bit of difference in their outcomes 20 years down the line, which is the Wisdom trial. I will promise you, I don't know, but I sure as heck wouldn't be promoting it until I had that data.
Have any of you computed polygenic risk scores on yourself? If so, why? Why not? And has it changed your behavior or medical therapy? Rob.
Yeah, so I have. So and so it has to do with obesity. So I'll just give you an anecdotal example. So about 12 years ago, my weight was 250 pounds. And these scores just came out, and so I thought, well, okay, so there's a reason why I'm 250 pounds. Maybe I can explain it based on my polygenic risk profile for obesity. So 23andMe was offering these, but we also were doing them in the lab as well. So I ran it. So it turns out I have a high polygenic score for obesity. But then, we looked at it, and basically then going from the, so the lowest decile to the top decile, the most that explains on the population level is around 3 kilograms. So about 6.6 pounds from lowest to highest, right? So I'm now 180. And so that was entirely due to the fact that when I turned 50, I just became like what I'm telling my patients to do. I started to try to adopt that myself and to try to be a better role model and better example for my patients myself. So again, these polygenic scores are telling you something. Most of that, and I think in extreme cases, they're really telling you something, but I think for most people, the genetic propensity, but can be overridden by lifestyle modification.
Thanks, Rob, for sharing that. Ruth, Steve?
I haven't changed my behavior. It would take more than a polygenic risk score, but no, I haven't done it for various reasons. I mean, these are good questions to ask. I mean, one of the questions sort of to ask is, at what point do you do the test, whether it's a mammogram or, I mean, people say, we're over-treating breast cancer. Should we change the treatment or should we change our screening protocol? In other words, once you've got the test done, these are sort of the critical, once the test is done, you're under kind of an obligation as a clinician even to act upon it. The question, I think the critical question as a community is, do you do the test in the first place? Like a very good example of that would be PSA. A person, I don't advocate for it, but if a guy has a high PSA, I'm not going to tell him to go home and forget about it. Similarly, I don't advocate for mammography, but if a woman has an abnormal finding on a mammogram, I would recommend that she has that, surgically evaluated and removed, even though I'm not sure that mammography is a good. So, the question I think is very much do we test or not test, rather. I think many of us feel obliged to follow up on the abnormalities.
Ruth?
Actually, I have not, because our studies are elderly people without heart disease or young people with heart disease. So at least the time we were running those chips, I didn't fall into either of those categories, so I haven't been genotyped. I guess I should be.
Any other questions from the audience? Yes, Steve.
So this is, I think, to all of you and also to Andrew. So Ruth had mentioned that the UK Biobank is now doing whole genome sequencing on the entire cohort, essentially. I think they have 50,000 done already. So can you offer your opinions of if maybe things are just too premature right now after a more comprehensive genomic data set is available and the GWASs are redone? Will the PRS constructs inform more on your phenotypes, but also other ones? And that's where I want to hear a little bit more from Andrew too, because I know he works in this area. So is it going to get better when there's more genomic data?
Yes, I mean, to the extent that, because, as you say, incorporating the rare variants, they have a large effect size with the others. I think, the attraction at the moment of a polygenic risk score is that basically you can run it on a chip, you can do the analysis very quickly for less than $100, even with the help of the bioinformatics team that does the imputation, et cetera. So it's very feasible. Once you're dealing with whole genome data on all of those individuals, I mean, it will be done. It is not as easy at the moment. Yeah. I mean, it's a very good point. I mean, but the UK Biobank is going to be a fantastic resource, and once again, a European population mostly, but not entirely.
Rob?
Yeah, no, I'm really the only issue is like computing capacity is like going to but I'm sure there's going to be a lot of new information, new models that are going to come out of having, that depth of information on that many people. But I think it becomes a challenge. Like you're almost going to have to, like, you can't store that much analysis. You have to almost throw it out each time or something. So but I think, I'm sure something's going to come out of it.
Yeah, I mean, Compute Canada has been an enormous resource for us, and we can store terabytes of information. But even with that resource, there are times when we don't have access, et cetera. So if we're going to deal with whole genome data sets on basically hundreds of thousands of individuals, that's going to be quite a challenge.
Steve, anything to add? No. I think we're... No, I'm the moderator. I have no comments. I think we're going to thank the three panelists and presenters for a great presentation and discussion. If you just want to stay there, I'm going to introduce Professor Melanie Wooden. She's the Dean of Faculty of Arts and Sciences at U of T. She's going to provide some closing remarks.
So thank you so much for inviting me to provide some closing remarks. And I just wanted to begin by thanking this afternoon's panelists, as well as the speakers earlier today, and the engaged audience for making such an outstanding inaugural CANSSI Ontario Research Day. What a great start. So yes, exactly. The Faculty of Arts and Science in the University of Toronto is home to the departments of Computer Science, Statistical Sciences, and Mathematics, as well as many data-intensive initiatives that range from disciplines such as the biological sciences all the way to the digital humanities. So the role of CANSSI and CANSSI Ontario in the Faculty of Arts and Science is really profound because it really is wide-reaching across our disciplines. And I think it's for this reason that Arts and Science is really ideally positioned to host CANSSI Ontario, because it has probably a pivotal role to play as a foundation for training and dissemination of knowledge in the data sciences and statistical science across not only the faculty, but the University of Toronto, its affiliated hospitals, and Ontario. So the programs in CANSSI, which the faculty is supporting, CANSSI Ontario with the support of CANSSI National. You've heard, I hope, a little bit about today, and these includes things like top-up funds for postdoctoral awards, access grant so that you can access large data sets, seminar and research funding series. So we want you to encourage you to take advantage of these. While CANSSI Ontario is housed in arts and science, it's really a resource center for the university and the affiliated hospitals. And I think one anecdote I take away from today is maybe an observation, and I'm sure many of you will have experienced this before. It's that you've gone to a conference, sometimes halfway around the world, only to come away thinking that some of the most impactful conversations you had were with people from your own institution, or somebody at the building next door, or at a university just down the highway. I see some people chuckling about that, so maybe that's happened today. And I think that's a real value of what CANSSI Ontario is doing, and CANSSI National, and certainly today, is that we're bringing people together who have shared interests, but may approach it from different disciplinary perspectives. And so it's really a fabulous opportunity to engage in those conversations. And at a time when travel is somewhat restricted, we need this more than ever. Yeah. So I'd like to say that certainly within my office in arts and science more broadly, and I think, or I hope all of you that are here today, we're certainly incredibly impressed with what CANSSI Ontario has been able to achieve in such a short period of time, which has certainly got us thinking very ambitiously about the future of CANSSI Ontario and what it can achieve. So I encourage you to think about how CANSSI Ontario can be a resource to you and the community across Ontario, and share your feedback with Lisa and her team so that we can make sure that we're supporting CANSSI Ontario in a way that really meets its goals and objectives. And with that, I'd like to really personally, and I think on behalf of everyone, thank Lisa Strug very much for everything she's done to get CANSSI Ontario going to such a fantastic start, and for today. And thank you all very much. Look forward to next year.